Yeah, to kickstart this last section off, we have Norrapat, who will share with us his work on the novel anticoagulant drug known as Variegin.
So a lot of the work in my lab is focused on looking for animals such as snakes or ticks, trying to isolate and identify bioactive molecules from their salivary gland or their venom gland. And subsequently, we try to develop some of these molecules into therapeutic drugs. So the molecule that I'm working on is an anticoagulant peptide called Variegin, which we previously isolated from the salivary gland of the tropical bond tick. And our plan for this peptide is to develop it as a treatment for ischemic heart disease. So ischemic heart disease is a leading cause of death worldwide, killing millions of people each year. So this is basically a huge problem in many parts of the world, and it's a problem that we want to tackle. So what is ischemic heart disease? As you grow older, there will be an increase in fat deposition in the vessel wall of your coronary artery, which is the major blood supply to your heart. And over time, this increase in fat deposition will form a plaque, which upon enlargement will basically reduce the hole inside the coronary artery for the blood to flow through. So what happens is, as this plaque enlarges, and under high blood flow and under high blood pressure, this may lead to a plaque rupture. And upon plaque rupture, this will expose certain signaling proteins to the blood system, resulting in the formation of this blood clot. Now, once formed, this blood clot will then block the blood flow to a large part of the heart, and depriving the heart of a lot of its needed oxygen, resulting in a heart attack. So, some of the risk factors that may increase your chance of developing heart disease or a heart attack may include excessive smoking, overeating or obesity, being angry a lot of the time, or simply just getting older. So when a patient comes into the hospital with a heart attack, what happens is the cardiologist would basically perform a procedure called PCI, or percutaneous coronary intervention. And during this procedure, the cardiologist would insert a catheter into the coronary artery in order to disrupt the clot and for subsequent stent placement. So this metallic stent, once placed inside the coronary artery, will basically keep the artery open and restore blood flow to the heart. So the issue now is that upon stent placement, this may actually result in even more plaque rupture. On a molecular level, what's happening is after plaque rupture, a lot of the proteins, such as tissue factor or collagen, are now exposed to the blood. And this will initiate the blood coagulation cascade, which in turn will activate this enzyme called thrombin. So when activated, thrombin can convert soluble fibrinogen into insoluble fibrin, which is basically a major component of blood clots. I took so long to make that. So in a young and healthy individual, the activation of this cascade is actually a normal wound healing process. But in an individual who has heart disease and also underwent stenting, this activation of this cascade may actually lead to the formation of a blood clot within the stent. And if a blood clot forms again within the stent, this may actually lead to a secondary heart attack. To prevent this, what we need to find is a very potent anticoagulant drug, which can inhibit the thrombin enzyme and prevent basically prevent the clot formation inside the stents. Now, since this drug will target the coagulation cascade, we would need, the ideal would be to find a drug that can both prevent clotting inside the stents while having a minimal bleeding risk. Now, the big question is, where do we look for these anticoagulants? Traditionally, the drug industry has been reliant on combinatorial chemistry to create large libraries of small molecules for high throughput screening. And although this method has yielded relative success in the past, a lot of the libraries that are generated are actually very limited in the number of molecular scaffolds available. Other methods such as structure-based drug design or in silico docking also suffer from quite a number of limitations. Mainly, one of the main issues being finding a molecule that in your computer simulation will bind to your target protein, and translating that molecule to function as a drug in animal models or even in humans. So instead of using some of these more troublesome techniques, let's say, our idea was basically to look for molecules which have already been selected by nature to perform their function. So ticks and leeches, for example, will produce anticoagulants in their saliva in order to help obtain its blood meal. Snakes, on the other hand, will produce anticoagulants to help subdue its prey. And the idea that we have is to basically search for some of these natural anticoagulants which have already been evolutionarily selected. In fact, there's quite a number of therapeutics out there in the market which were originally derived from the toxins of different venomous animals. One famous example is Variegin, which is a very potent anticoagulant derived from the medicinal leech. And a second famous example is a type 2 diabetes drug called exenatide, which was originally developed from the Gila monster. So I guess the overall take-home message here is that the toxins from a lot of these venomous creatures can provide a rich library of very potential therapeutics. So the molecule in which I'm interested in is an anticoagulant peptide called Variegin, which we originally isolated from the salivary gland of this tropical bontic right here. We found that the Variegin peptide can act as a high affinity thrombin inhibitor and also a very potent anticoagulant in our animal models. So to study the effect of our Variegin peptide, we employ two different animal models, first being the rat carotid artery thrombosis model, which we use to study the efficacy or the effectiveness of our peptide to prevent clotting in a blood vessel. The second model that we use is the tail incision model. And we use this model to basically study the safety effects or the potential bleeding risk of our peptide. So for our first model, the efficacy model, firstly we have to surgically isolate the carotid artery from the rat's neck. So after exposure we can place a Doppler flow probe on the carotid artery to measure the blood flow. And subsequently, we can use a filter paper which is soaked in a chemical called ferric chloride to cause an injury on the blood vessel. So the idea is that the ferric chloride chemical will induce the formation of a blood clot within the carotid artery. So after this, we can use the flow probe to basically measure the blood flow over time after the injury, and subsequently measure a value called the time to occlusion, which is basically the time until the blood flow comes to zero. So basically, with more anticoagulant inside our model, this would result in an increase in time to occlusion. For our safety model, what we basically do is we basically take a scalpel blade, make an incision on the rat's tail, then we use a filter paper to dab the blood from the wound, and after this we can measure the bleeding time, or the time until the bleeding stops. So we use this model to basically study the potential bleeding risk of our drug. Now, by using these two models, we can assess the effect of our peptide in comparison to heparin, which is the current drug in the clinic being used for PCI. And if you look at the data for heparin, you can see that with increasing doses of this drug, there's a gradual increase in the time to occlusion in our efficacy model. And this increase is followed by an almost proportional increase in the bleeding time. This is in contrast to our Variegin peptide, where a dose of the Variegin peptide that gave a similar antithrombotic anticoagulant effect in comparison to heparin, those doses actually gave a much lower bleeding rate. So what this data shows here is that the Variegin peptide has a much wider safety window. The Variegin peptide has a much wider safety window and basically can potentially be a safer drug for use in the clinic. Our future plan for this drug is to basically perform different animal models to prove its efficacy for different indications, and hopefully within a few years we'll be able to push this drug to clinical trials. Thank you.